and welcome to the Amplifying Scientific Innovation podcast. I'm your host, Dr. Sophia Onoye, founder and CEO of the Sophia Consulting Firm, a life science marketing and communications consultancy that was established in New York City with the goal of amplifying scientific innovation. The goal of this podcast is to showcase scientific innovation stemming from global life science companies through conversations with senior leaders who share their unique leadership journey, corporate vision, and industry outlook. My guest today is Dr. Doug Manion, CEO of Kleo Pharmaceuticals, a unique biotechnology company that is developing next-generation bispecific compounds designed to emulate or enhance the activity of biologics based on the groundbreaking research of its scientific founder, Dr. David Spiegel at Yale University. Doug is an experienced senior leader with a demonstrated history of working in the biopharmaceutical industry. He spent over 11 years at Bristol Myers Squibb, BMS, and approximately 4 years each at DuPont Pharmaceuticals and GlaxoSmithKline, GSK. Doug obtained both his Bachelor of Science degree in Civil Engineering and Doctor of Medicine degree from the University of Ottawa. In addition, he completed a postdoctoral fellowship in retrovirology from Harvard University. Doug and I met in person at the Kleo Pharma headquarters in New Haven, Connecticut, following an introduction from Vladimir Kurik, CEO of Biohaven. We discovered a mutual passion for immuno-oncology and the state of Connecticut. Welcome to the show, Doug.
It's great to be here. Thanks for having me.
It's my pleasure. So my first question is my favorite for several reasons, but I will start by asking you, what is your definition of scientific innovation?
Yeah, so I love that question. It's a question about which I've been thinking my entire professional career. And my answer to the question has mutated over time, but now it's pretty firmly grounded in the following. Scientific innovation is having a novel idea to do things differently, but also to have the means by which you can actually get that concept realized pragmatically so it has some impact on other people's lives. So there's a ton of people have good ideas. It's not sufficient to have a good idea. You have to have a good idea, and then you have to have a plan to actualize that idea, which includes especially, not uniquely, but especially in the private sector, it also means that you have to convince people that they should put their money behind that idea and that they should use their money to create a team that can actually efficiently convert that money and that idea into something that's going to pragmatically improve people's lives. And there's a million people who they see others doing. I mean, the classic would be Apple, right? Where there's nothing unique about Apple. I mean, Apple makes phones, and Apple makes computers, and Apple makes other things. Apple just does it better than other people.
Right.
And in fact, initially, it was believed that Apple didn't do things better than other people. Apple, when they came out with the Mac computers, they were deemed to be way too expensive compared to PCs. Initially, Apple was floundering. Steve Jobs was fired because people did not believe that his vision could be actualized, and he dogmatically stuck with his vision. And Apple now is $1 trillion company. It's one of the richest companies in the world. And another thing about Steve Jobs, though, is his innovation. One of his biggest innovations is a thing called the graphic user interface, or GUI. So GUI, that was not invented by Apple. It was invented by Xerox. And Xerox invented this. They had an R&D facility where people were allowed to just kind of do fun stuff. So they were doing this amorphous research in terms of computer technology. They came up with this idea of a graphic user interface, which the graphic user interface is what we now all use with the point and click for all of our devices, PDAs, iPads, you name it. Xerox saw zero value in the GUI. And Steve Jobs saw this as the future of computing. And it was Steve Jobs' one realization that that was the case, but also his ability to just wrestle the IP away from Xerox, form a company around it, is why Apple is Apple. So a long-winded answer to say that A good idea that doesn't get realized is like a tree falling in the forest with nobody around to hear it. Some of the greatest innovations are not that clever, but they have follow through. And I can give you lots of examples, but I don't want to bore you.
No, I love your definition around pragmatic improvements and also differentiation. And I think one of the things you alluded to is, as a leader, you have to have a core belief in what you're doing, even if other people don't buy into it. So I like that you captured that in your definition. So my next question, you've done quite a lot, and you should be very proud of yourself. But what is the one thing that you will consider to be your most significant professional or scientific accomplishments today?
So it's another great question. I love talking to you. So when you get a little bit older, you get a little bit philosophical. And so my role in the scientific innovation ecosystem has always been much more that of the engineer than the designer. Mm. So I am not a particularly innovative scientist. I'll be blunt. That's not what I'm good at doing. I'm an engineer. You mentioned I'm both an engineer and a doctor. I use my engineering every day, because engineering is basically just problem solving. And engineering is basically taking good ideas and realizing them pragmatically. I was actually a sewer engineer, so there the problems are pretty simple. You want to get bad stuff from here to there, and you want to find a cost and time efficient way to do so. So my contribution has always been that I have, I think, a better than average radar when it comes to seeing good ideas that are worth funding and realizing, and then to bring together a highly innovative, diverse team to actually make that happen. And what I find in science is you have a lot of very, very smart people, but you also have a lot of very, very strong personalities. And left to their own devices, many, many scientists, academics, private sector scientists in companies will work on their own because they're not naturally collaborative. You have to kind of collaborate. And a lot of it is just their belief in how right their science is and their kind of unwillingness to compromise that science. But at the end of the day, it takes a village, it takes a team to turn anything into reality. And in terms of drug discovery and development, it takes a team of hundreds, if not thousands of people. And it takes years, if not decades, for it to actually happen, right? So what I did throughout my career, starting in academia at Harvard, and then subsequently at the various companies I worked at, and now at Kleo Pharmaceuticals, is I get together very, very strong, bright teams, and I find ways to make them work with the eye always being kept on the prize, and the prize is to create drugs that are going to have a macroscopic impact on people. So every drug that I've been involved in that's gotten to people has had multiple near-death experiences. I think drugs are almost like cats. They have 9 lives, and if you give up too soon, you won't make it. So the first drug I worked on was a drug called Sustiva. Its generic name is Afara. So the company called DuPont Merck Pharmaceuticals. I left academia to work on it, and it just in the first 6 months I was there, even though it was in phase 3 research, it had multiple near-death experiences, So, and it wasn't me, it was the CSO of the company, a guy called Paul Friedman. He refused to let that drug die. He knew it was going to be a good drug for people, and he willed it across the finish line in terms of FDA and subsequent global approval of the drug. But it took a really talented team of people that all shared in the same belief, and that didn't, kind of rest on their personal dogmas. We're willing to kind of listen to the other points of view and come up with solution sets that they would not have come up on their own. And we solved a tremendous number of problems that lesser human systems would not have been able to. And I worry sometimes, how many drugs are currently in kind of the trash heap of scientific history because there just wasn't the right backing, there wasn't the right champion, and there wasn't the right team collaborating with the right diversity of scientific abilities to solve these really tough problems.
I like how you've combined that sort of practical knowledge of civil engineering with a technical proficiency in medicine as well as molecular biology. I think that has played a very important role in your journey today. So, but then I'm curious then, why did you choose to switch from your career in virology and infectious diseases in big pharma to cellular immunotherapies at Kleo Pharmaceuticals?
Yeah, I can't remember who said it. It might have been MacArthur or Eisenhower who said, the first thing that you do in war is you throw away the war plan. So I had these big plans for my career, but life is what happens when you're making plans to do other things. So I went into med school very specifically to become an AIDS doctor and an AIDS researcher, and I was very emotionally committed to having a macroscopic impact on HIV/AIDS. So that was 1983. I ended up studying literally for 14 years. So my time in Boston was mostly as a postdoc. And so 14 years later, at age 37, I finally got a real job. And my first job actually was helping to develop what turned out to be one of the greatest AIDS drugs of all time. And that was 1997. And you may remember 96, 97 is when HIV went from being a almost universally fatal death sentence to a controllable disease by using a drug cocktail called highly active antiretroviral therapy, or HAART. Well, the drug I worked on, Cystiva, was a critical element of the first wave of these cocktails. And because it was a one pill once a day offering, it was easily co-formulatable with other similar pills. So in collaboration with a company called Gilead, we formed a drug called Atripla that was pivoting to other infectious challenges. So I began working on hepatitis B initially, and then hepatitis C, and this is in 2005, 2006, 2007. There was a Manhattan Project in the world in terms of curing HCV. I led the effort of BMS. BMS actually didn't win that race. Gilead did, but we made it really important contributions. But already by the mid-2000s, I was not HIV specific in my infectious diseases work. And then because I had spent some of my time at Harvard learning about the immune system's response to HIV, which we now call immunovirology. I had expertise, especially in T-cell immunology. And it just happened at BMS, there was an opening for the head of clinical immunosciences, overseeing their efforts on lupus and rheumatoid arthritis and other autoimmune diseases. And so because of a belief that I was a good leader, because of my history in terms of immune expertise, was given the opportunity to lead the immunoscience group, and then I did a good job with that. The next thing you know, they added on neurology, and then they added on global biostatistics, and then they added on all of Asian R&D. So I had in Japan, Korea, Taiwan, and China for 6 years at BMS. And they just kept adding things on. They realized that I was a versatile leader, and my secret sauce wasn't deep expertise in any therapeutic area. My secret sauce was being able to get people to synergize, including very senior, very strong-willed scientists, right? So basically, by the end of my stint at BMS, I was overseeing all drug development and medical affairs for therapeutic areas other than oncology globally, both Yervoy and its sister drug, Opdivo. Opdivo was an anti-PD-1 monoclonal antibody, which is very successful. So I actually oversaw the approval of those two drugs throughout Asia. So I had a front row seat to the first wave of really large-scale immuno-oncology therapy. Immuno-oncology had been around for decades, but it never really got a foothold because they didn't have the right mechanisms. But checkpoint inhibition turned out to be an incredibly valuable intervention. As you know, just 2 years ago now, the discoverer of both the CTLA-4 pathway as well as the discoverer of the PD-1 pathway shared the Nobel Prize for Medicine. Jim Allison, who I know was actually the recipient for CTLA-4, which is really exciting. So, long-winded answer, when I was leaving BMS, there was a huge investment going into immuno-oncology. I was very committed to helping grow out the biotech sector in Connecticut. I think Connecticut should be doing much better than it has been doing in terms of biotech. And so, the company Kleo Pharmaceuticals, I thought that they had a really unique slant on intervening in cancer through modulating the immune system. And so I met with David Spiegel and with Roy Preeb, the founders, in the spring of 2017, and it just seemed like an obvious fit for me. I joined in May of 2017.
Oh, that is great. I think from your story, I hear about the power of serendipity and being good at what you do, being able to leverage skill set from one disease state to another. I think that has been quite good, I guess, influential in making you who you are today in some ways, right?
I think that's beautifully put. And I think in life, you got to kind of figure out, do you want to just go ever deeper in one narrow area of expertise, and there's value in that? Or do you want to be more of a generalist who can adapt to a bunch of different kind of scientific applications? And I chose the latter, and it has worked out great for me. Now, again, I think part of what I bring to the table is I'm good at finding those people with very, very deep. So for instance, the CSO of my company, Luca Rastelli, extremely experienced immuno-oncology biologist, very, very kind of narrow but profound area of expertise. So part of my value is teaming up with him and David Spiegel to leverage those experts. I mean, David, of course, is a genius when it comes to synthetic and medicinal chemistry. So my helping them synergize, my helping us leverage their deep area of expertise, complementing their depth of expertise with other capabilities, because you need a whole bunch of capabilities to discover drugs, is kind of what I do.
That's great. And I'll be curious to know how those capabilities have evolved amid COVID-19. So can you provide a top-line overview of ongoing work that is being done at Kleo Pharmaceuticals, especially as it relates to COVID-19 and, of course, your core in novel immunotherapies?
Yeah. So the COVID-19 story is a great one and underscores a lot about what I love about Kleo. So Kleo, we are a synthetic chemistry company, right? So our central premise is that if you're smart from a chemistry's perspective, you can do with synthetic, putative small molecules, peptidic or non-synthetic, small molecules, what others can only do with large biologicals like antibodies or cell-based therapies. So the beauty of synthetic chemistry is that you have at your fingertips an incredible diversity of kind of chemistry. Way more natural amino acids, right? So God only gave us 20 natural amino acids that are strung together in proteins, right? So you're limited by definition in terms of the chemistry. But for synthetic peptides or even non-peptidic chemistry, you're basically almost unlimited. You're limited only by your imagination, right? So a couple of things kind of set us up for the successes that we're now seeing with COVID-19. The first was that we had a couple of years ago gotten the idea that we should look at our synthetic chemistry approach to see if there wasn't a way for us to permanently chemically modify antibodies to make them better. And that evolved into a platform that we called mating, or monoclonal antibody therapy enhancers, which is technically now a misnomer. But we could take any antibody therapeutic, be it a monoclonal antibody like Erbitux, Rituxan, Darzalex, elotuzumab. So we could chemically engineer those antibodies to either redirect them against other antigens or to confer to them different activities. So we could take, for instance, off-the-shelf Rituxan, and we can turn it, which doesn't naturally turn on killer T cells, and we can basically turn it into a killer T cell binding and activating targeted immunotherapy. So Luca had a very good idea. So anyways, with COVID-19, they were able to elucidate this surface protein called the spike protein. And the beauty of our small molecule chemistry is that we could very rapidly create very high affinity small molecule binders once we know the protein structure of a target. So we immediately began a COVID-19 program with the intent of finding binders that we could then bind to antibodies, IVIG, to convert healthy volunteer IVIG into a hyperimmune globulin mimic that basically creates a synthetic version of what we all are hearing about now called convalescent serum. So convalescent serum, you take the antibody, you take the serum from people who survived COVID-19, you measure antibodies, make sure they have some against the virus, and then you give that serum to people either to protect them from getting the virus, or if they already had the virus, to lessen the severity of the disease. So this approach has received emergency use authorization from the US FDA about a month ago. And there's been a positive finding for it. But the issue with convalescent serum is you need to find enough people who have had COVID-19, who are one, willing to give up their serum repeatedly, and two, have enough antibodies in their serum to make it worth doing, which is a challenge to do. So we can get around all of those challenges because we can take and it's very easy to get healthy volunteer immune globulin, and we can convert it into very high titer, very high affinity anti-COVID-19 convalescent serum-like therapy. So clever is this? And again, this wasn't my idea. This was Luca's idea, but I helped to make it come to fruition. We basically pitched this to the American government and to the Gates Foundation. Both of them loved it. This was in the March timeframe. And then as of July 7th, we've announced that the Gates Foundation is funding initially with a grant of $5 million, all of the work on this hyperimmune globulin mimic, or HGM program. And the intent would be, as long as the science continues to progress, and it's been progressing really well, that they're going to pay for everything.
Wow.
We have line of sight to first human dosing either very late this year or very early next year. And we think that this could be a game changer in terms of COVID-19. I mean, everyone is hoping that the vaccines are going to work. Everyone is hoping that the monoclonal antibodies are going to work. But this is a completely unique mechanism of action that is almost halfway between vaccines and monoclonal antibody therapeutics. And we think that this could be an extremely important therapeutic approach to COVID-19. And, Gates Foundation, the American government seem to agree. And the further beauty is that it's not just specific to COVID-19. We could do this for any disease where hyperimmune globulin has shown activity, and there's many such diseases. Respiratory syncytial virus that affects young children and older individuals, hepatitis A, hepatitis B, rabies. So in fact, we believe that this approach could be what I call a disruptive technology for the hyperimmune globulin marketplace.
Wow. Oh, first of all, congratulations. That's a lot of good news. And it's not just the news, it's about what you had said earlier regarding scientific innovation, right? Pragmatic improvements, and the fact that Kleo Pharmaceuticals is evolving its platform at a time of great global need. It is demonstrative of really your brilliance as a leader and of course, your great team of scientists. So now
I'm brilliant as a leader, but if I do keep the cats, I heard cats better than average. Let's put it that way. All the credit goes to the visionary scientists like Luca and David, but you need an engineer. I remember so I'm Canadian, and for the 1976 Olympics, they had this worldwide architecture contest, and they got these architects to design the Olympic Stadium. And the winning design was a brilliant design by this French architect. The only problem with the design, and it won the competition in like 1970, so they had 6 years to build it. The way it was designed, it couldn't be built in less than 50 years. Because it required consolidation of the soil. At the time, there was no way to compact the soil fast enough to be able to build it. So you may remember the Montreal Olympics were held in an incomplete stadium. And it took them another decade to finish the stadium because the engineers had to figure out how to do it, right? So you can have the greatest, most visionary idea, but unless an engineer can actually make it happen, it ain't gonna happen.
Well, that's great. So I think I know the answer to this question already based on your response, but I'd like to hear it in your own words. What is your vision for Kleo Pharmaceuticals?
So always, the vision, our North Star for anybody who's in the pharma industry, biotech, big pharma, whatever, if your North Star is not helping patients, you're in the wrong business and you should be doing something else. So I went into med school to have a macroscopic impact on HIV and AIDS. And my personal mission in life is to continue to have macroscopic impact on areas of high unmet medical need. And the vision for Kleo is to leverage its very unique way of creating and developing therapeutics to have as big an impact as possible on as many diseases and people as possible. When I joined the company in May of 2017, I had them focus all of their efforts only on immuno-oncology because we're a small company and you can't be too spread out. But already we're back into infectious diseases because of COVID-19. And there are many, many other diseases that could benefit from our technologies. So at the end of the day, helping as many people as possible. From an investor perspective, money is a surrogate marker for helping people, right? So we're not in the business of making money, we're in the business of helping people. But trust me, if you help people medically, the money will come.
Absolutely. Very well said. So my next question for you is, as we switch to a macroscopic view, is how do you think the biotechnology industry has changed in the past 15 years? And I guess the follow-on to that is, what are some loopholes?
Yeah. So there are many kind of macro trends, but the one that I've seen that is the most important, and I think probably the best, is that very large pharma has pivoted from going away from the not invented here approach to seeking innovation anywhere they can find it. So it used to be that most big pharma companies, Merck was the prototype for this. If it wasn't invented by a Merck scientist, it wasn't worth doing anything with. So now there are entire large companies that aren't even doing any of their own discovery work, where basically they outsource all of their discovery to these highly innovative entrepreneurial companies like Kleo. And many of these big pharma companies also have venture arms where they have money available to help support entrepreneurial biotech companies like me to basically percolate these good ideas that then get absorbed into big pharma because you need a lot of money and capabilities to execute against it. So that to me is really virtuous, because as soon as you attempt to innovate in a closed system, I feel you're dead in the water. You get into these echo chambers. Big pharma, for instance, it's a very common thing in big pharma that if you had a success with a certain approach, you falsely believe that approach is going to work again.
Yeah.
And you can't predict that. And big pharma has gotten into ruts, very frequently gotten into ruts. So what you want there to be this diversity of thinking, and you want open to, some people think that what we're doing at Kleo is crazy. To which I say, well, just watch. And others, the Gates Foundation being one of them, doesn't think we're crazy. They think that we're genius, right? So that's virtuous. That is a free market economy that is basically rewarding innovation early on, as opposed to stifling innovation. And to me, the more that all of the elements of the ecosystem are synergizing, public sector, private sector, academia, even kind of non-scientific groups. I mean, certainly venture banks and the like. The more that people are collaborating, the greater the likelihood that you're going to come up with better ideas and the funding necessary to realize them for pragmatic use. Most importantly, to help people. So there is a virtue, for instance, to the whole venture capital scheme that drives innovation in the medical field. These are super smart people that put their money where their mouths are, and that's good. Academia is much more cash-strapped and much more limited.
Right.
There are still barriers in terms of private enterprise interacting with academia that need to be overcome. I think it's getting a lot better, but there's so much more work that could be done to make it easier for us to work with academics. Academic institutions have a lot of restrictions in terms of what their people can do, and some of them are good and some of them are less good. So anything that you're doing to accelerate the collaboration across sectors, across scientific disciplines, the better off we're all going to be. All boats rise.
I love the emphasis on collaboration. I think that is really key. And for my next question, I also like that question for you because of your background in infectious diseases. So I want you to be as honest as you can be, but I'll be curious to know, what could we have done differently as an industry to mitigate the impact of COVID-19?
Again, a great question. I'm gonna kind of harken back to HIV. And the good news is that we did not repeat the errors of the past. But in HIV circles, after the cocktail was invented in the mid 1990s, it was only accessible to the richest countries of the world, and even then, the wealthiest or best insured people in the richest countries of the world. So there was a movement to try to make those medicines available to poor people, and our industry stood in the way of it. There was this maniacal focus on intellectual property, and somehow if we allowed cheap drugs to be manufactured to be given, for instance, sub-Saharan Africa, that somehow those inexpensive versions of our drugs would flood the US market and everyone would lose their shirt and all hell would break loose. And so there was almost a lost decade where we couldn't get out of our way and we were fighting for things that weren't worth fighting for. And it was only really in the George W. Bush administration where they came up with a thing called PEPFAR, where they put money behind it and the industry got out of its own way and good things happened. So fast forward now to COVID, the really good news is that there's really good synergies between the public and private sector. There's really good, so for instance, NIH has put together avenues for small companies like me to be able to tap into capabilities. For instance, animal research facilities that are running COVID-19 models that we wouldn't have access to otherwise. The federal government, through Operation Warp Speed and other things, has massively invested in terms of at-risk infrastructure building, for instance, for vaccines and therapeutics. The philanthropy area, like the Gates Foundation, has been prescient in terms of funding, and I'll be blunt, funding companies like mine that wouldn't have gotten funding otherwise. So all of that is really good. What isn't happening yet is synergy between the private sector companies. I mean, we're competing as opposed to synergizing. I'm not sure if we're ever going to be able to stop synergizing. I will tell you, the answer to COVID-19 is almost certainly going to be similar to HIV, is that it's not going to be a single intervention. It's going to be an all of the above intervention.
Right.
So, I mean, the goal of the vaccine is 50% effective in the 50% of people who are willing to take the vaccine, right? So that means 25% effective on a population basis, right? So you're still going to need drugs, and you're going to need drugs, different phases of the disease, antivirals for when it's a viral-based disease, immune suppressants when it's an immune-based disease, possibly combinations of both. So the sooner that we are looking at multi-drug approaches, and that there's a vehicle by which companies can collaborate and not necessarily compete, the better. NIH, for instance, does have a master clinical trial that allows multiple drugs to be kind of put into this infrastructure. So that helps a bit, but it still isn't really companies synergizing, not quite yet. But it's way better than it was 20 years ago with HIV, I'll tell you that.
Oh, that's brilliant. I like the emphasis on collaboration. I think that is going to be key. And it's not just this sort of solution, just one solution, one answer. It's going to be multi-pronged. So I really like that emphasis that you placed. So my next question is easy. Is there any technology or company that you're currently excited about, and why?
Yeah. So we're really modular. And so there's a bunch of companies that are similarly modular. So one of them is a company with whom we have a collaboration, Peptidream.
Yeah.
Peptidream is one of the world's most clever synthetic peptide discovery companies. So they don't make drugs. They make components of drugs. And they have deals with a bunch of different pharma companies, including ours, where they are basically a peptidics discovery engine for us. Right? And then, for instance, the lead asset that we have going to the clinic, which is called KP1237, it's an anti-CD38 antibody recruiting molecule. Its CD38 binder is, in fact, a peptidream binder. Right? So I love these companies that part of their mission is to be part of other companies' solution set. Right? And then, so they're worth about $6 billion, publicly traded on the Nikkei. But all of their revenue generation is through collaboration with other companies. So I love the model and I love the science. So Peptidream is a great example of a company that I think is cutting edge in terms of where everything is going. They have 17 large pharma deals, and they are the synthetic peptide discovery engine for pretty much every large pharma company in the world. We're not a large pharma company, we're a small pharma company, but they serve that role for us as well. So that extremely high level science, but a business model that is fundamentally collaborative. So I kind of love that.
That's great. That's brilliant, actually. I think it takes both to be successful, not just science, but also the business acumen. And you mentioned engineering earlier, the practical element. So my next question, you sort of answered it in bits and pieces already, but what are some key consideration factors outside of, say, collaboration that you think that will be important for sustaining innovation in the life science industry?
Well, I'll state the obvious, that if there isn't a profit motive, it would be a huge break on the system, and this takes us down a very thorny path, right? So, drug pricing is a politically hot button issue. Were it not for the pricing scheme allowed in the U.S., there would be a significant blunting of investment in biotech. We just need to be blunt, because almost every other Western democracy has some level of price controls.
Yes.
And so basically, U.S. pricing and the U.S. market subsidizes R&D for the rest of the world. So the U.S. consumer is basically paying for 80% of all medically related or pharmaceutical R&D around the world. So that's unfair to the American people. And if America goes the route of price controls too, then it just means the amount of money available for pharmaceutical research is just going to go down. And by definition, it's going to hamper innovation. Right? So my belief is that you can have sane pricing whilst without killing the goose that lays the golden egg. And that is by making sure that there's the right emphasis on drug purchasing. Right? So for instance, in the United States, still many people don't have access to drugs because of access issues, not because of pricing issues. Right? So if there's more uniform access to innovative medicines, then that alone would be sufficient. Even if there was some curtailment of pricing, you'd make up for it on volume, right? So those are the types of things that I'm hoping that we're going to get straight moving forward. And that should be true for pretty much every jurisdiction. There should be some free market ability for companies to set their initial price in markets. There should be some sort of a volume price negotiation that's ongoing. So in Japan, the way it works is you set an initial price like you do in the US, and then there's almost like an algorithm of adjustment of price based on volume of sales. And it's predictable for the company, right? More money up front, less money, as time goes on. But it actually works for everybody, right? So that's a scheme that could be considered by other jurisdictions. But I think biotech pharmaceutical investment is always going to, even if there were more price controls in the U.S., it is still a more lucrative area than many other areas of R&D. So it's never going to completely go away, but one could predict that the smaller the pot of money that could reward innovation, the less money will be backing innovation. It's just a free market, right?
Yeah. No, I think you make some excellent points. My personal belief is that those collaborations that you mentioned, as well as emergent technologies like artificial intelligence, will help to reduce the inefficiencies in the drug discovery process, which will, of course, play a role in drug pricing. But I'm sure we could, that's arguable to an extent, but I do believe that all those things sort of go hand in hand.
No, I agree completely. And one of it's interesting coming from virology. So one of the reasons why I went to virology because I hated HIV, but learning pretty quickly as I became a virologist is viruses are really simple. I mean, compared to other polygenetic diseases like cancer, by definition, viruses are monogenetic. The virus has a genome. We know exactly what the genome is. It could mutate over time. But for instance, HIV has a 9,000 byte genetic makeup. It encodes for a modicum only of proteins, and we know what those proteins are. So it's a relatively simple, from a genetic and protein basis, it's relatively easy to thwart a virus. Right? And in fact, most viruses in humans, you could predict how the virus is going to interact with humans based on modeling, based on in vitro.
I couldn't agree with you more, and I like that we're having this conversation because I think it will help to educate the public on why those drug prices are as presumably high as people say they are. So my final question is simple. Do you have any commentary or thoughts that you want to share? The floor is yours.
Only to applaud you and such podcasts. I think another thing that's really good, I'm of course, with being sequestered by COVID, we're finding new ways to entertain ourselves. I've become a little bit of a podcast addict. But I tell you, it's a great way to get kind of in-depth knowledge on a topic really quickly. Yes. And I think I give kudos to folks like you who have the vision to create these vehicles for people to better understand stuff. So it's an honor to speak to you. I, of course, love you. And I think that this is good. I really applaud you for making these things happen.
Well, thank you, Doug. It's my absolute pleasure. I've really enjoyed speaking with you. I think I could speak with you all day, but let's save the date for another future conversation. So I appreciate your time, and I definitely look forward to staying in touch.
Anytime you want me back, I'm just on the phone. All right. Bye, everybody.